Hi, this is the next nervous system lecture on nerve impulses, and the question today is, what causes a nerve impulse and how does it propagate down an axon? By the end of this lecture, you should be able to answer this question using all of the vocabulary words on the left. The ones on the right are there too, but they're going to be part of a separate lecture, so you're only going to get a very brief introduction to them here. So the content of this lecture is on how neurons are able to transmit impulses because of their resting membrane potential, which becomes depolarized and then later repolarized. And when they become hyperpolarized, the sodium-potassium pump fixes that so that they go back to their resting membrane potential. I'll also very briefly talk about synapses and neurotransmitters, but that is a topic for the next lecture. So to recap, most neurons that are in this unit that we're going to talk about are multipolar neurons. So here you have the soma, you have these nerve processes that come out of them called the dendrites, and the dendrites receive signals, and then the signals get transmitted down this other long nerve fiber called the axon, all the way to the axon terminals, where the signals are transmitted to another neuron. The axon will probably be myelinated, and when it does have myelin, that allows the signal to be more efficiently transmitted down the length. To put all of this in perspective, one neuron is connected to other neurons, which are connected to other ones, which are connected to other ones, and they all send signals from one to another, and they crisscross throughout the entire brain and down in the spinal cord, out through all of the nerves in your body. And so this transmission of information is what allows your brain to control basically everything that your body does. How do neurons transmit nerve impulses? Well, they have two distinct properties that make them different from any other cell in your body. First, they're irritable, so their special shape, their structure, allows them to respond to stimuli and convert those stimuli to nerve impulses. They're also conductive, which means that they're able to transmit nerve impulses to other neurons, muscles, or glands. Now, neurons, a lot of the times the stimulus originates from outside your body, and they come to sensory organs like your eyes or your ears or your skin or your nose and your mouth. And so neurons in those sensory organs will then convert those signals into chemical signals. Now, how do those neurons in the sensory organs then communicate with other neurons, and how do those neurons then communicate with other ones? It starts with a chemical signal that originates from the sensory neurons that are in your sense organs. So those chemical signals will be received at the plasma membrane, usually at the dendrites. And then that chemical signal is propagated as an electric signal all the way down the axon, and then it is transmitted to the next neuron as a chemical signal at the axon terminals. So we're going to start with a chemical signal, we're going to turn it into an electrical signal, and then we're going to turn it back into a chemical signal to send the signal to the next neuron. You'll also notice that I put the word propagate in red because even though it's not technically like a standalone science term, it's still a good word that we can use to talk about how signals go all the way down the axon from one end of the neuron to another. So it all starts here at the resting membrane potential, which is this term refers to the polarization of the neuron's cell membrane that makes the neuron both irritable and conductive. And the reason why you need to know about this is because you have to remember that nerve impulses begin at the plasma membrane. But they don't happen in any other cell in your body. And the reason why is because only the neuron's plasma membranes are polarized at rest. What that means is they have electrical charges that are uneven on both sides of the axon membrane. Here you'll notice we have these sodium ions, which chemical symbol Na+. There's a lot of them on the outside of the axon, where the axon is represented by this blue cylinder. So you have some up here, you have some more down there. You also have potassium ions represented by chemical symbol K+, and these are also positively charged, and they're inside the axon. So positive charges outside the axon, positive charges inside the axon. But then you also have these little red minus signs sprinkled outside and inside. Those represent negative ions, and they're present everywhere. And if you have more positive sodium ions outside the axon, then you have potassium ions inside the axon. What that means is the inside of the axon ends up becoming relatively negatively charged, and the outside of the axon ends up becoming relatively positively charged. So let me illustrate that a bit more. You're going to have negative charges both outside the axon, which I've labeled for you here, and you're going to have negative charges inside the axon. And you're going to have positive charges on both sides. So some of those positive charges are sodium ions, which, I don't know, let's say they're red, and some of them are potassium ions, which let's call them blue. It doesn't really matter. And then you have the axon membrane right in the middle, shown in yellow. So when the amount of positive charges are equal on both sides, then they're going to cancel out the negative charges and everything is neutral. But if some positive charges exit the axon and go to the outside, now all of a sudden you have more positive charges outside the axon than negative. And you're going to have fewer positive charges here inside the axon than negative charges. So the inside becomes overall negatively charged, and the outside becomes overall positively charged. And so that creates a polarity across the axon membrane, where the outside is positive and the inside is negative. And the reason is the outside has more positively charged sodium ions than the inside has positively charged potassium ions. Outside positive, inside negative. Outside has more sodium, inside has potassium. Just to clarify, there's going to be sodium ions and potassium ions both outside and inside, but in general, more sodium outside, more potassium inside. So now that the signal has been received, right? All this time the cell was at rest, and this was the resting membrane potential. It was polarized. But now there's a signal that's coming, and that's going to depolarize the axon. And so depolarization refers to when some event triggers an action potential in a neuron and enables the action potential to propagate down the neuron's axon. When this happens, we call it a nerve impulse. And so I'm going to use the word action potential and nerve impulse interchangeably. Let's just go ahead and call them synonyms. So here's what that looks like. You'll notice that at rest, this neuron's axon is positive on the outside, negative on the inside, until all of a sudden, here's an action potential, and you're getting a depolarization of the membrane, where for a very short amount of time, part of the axon membrane flips. It becomes positive on the inside and negative on the outside, and that positive inside depolarization travels from the dendrite side of the axon all the way down to where the axon terminals are. Here's how that happens. So for the resting nerve fiber, we have these green circles which represent the sodium ions at a high concentration outside, and the blue circles which represent positively charged potassium ions at a high concentration on the inside. But when the message is arriving, as you can see here, when the message arrives, it causes these channels to open in the axon membrane, and that allows sodium ions to rush in. And then when the sodium ions come in, that makes the inside of the axon positive, just for a very brief moment in time, until those sodium ions eventually go out again. It's analogous to if you had a balloon, like one of those balloons that you use to make balloon animals, and if you stepped on it, it would push the air all the way from this end to the opposite end. And so that's analogous to how the sodium ions, that positive charge, gets propagated or pushed all the way down the length of the axon. So here's another depiction of that. We have sodium ions that were outside, but because this blue sodium channel opened, now the sodium ions can rush in, which makes the inside of the axon very, very positive. Um, okay, and here's another depiction of that at the bottom. You can see that we've got a lot of green sodium ions outside the membrane. And then all of a sudden, sodium channels will open and the green sodium ions rush in. And for a brief moment in time, the inside of the axon is going to have as much sodium as the outside does. So the inside becomes positive until you close the sodium ion channels, and then the sodium starts getting pumped out again, and then eventually you're going to have sodium back outside. Here's all of that in words. So starting with panel A, you've got your resting membrane potential. Nothing is happening yet. Positive outside, negative inside, just the way it should be. But a stimulus depolarizes the neuron's membrane by opening up sodium ion channels or sodium ion gates on the membrane, which allows sodium ions to rush inside. And that's panel B right there. So stimulus initiates depolarization when the sodium ions come inside. And for a very brief moment in time, the inside is now depolarized. So that initial exchange of ions is a local depolarization. It makes the inside more positive than the outside. And then that depolarization starts an action potential in the entire neuron where now that the inside is positive here, that's going to trigger more sodium channels to open, so more positive charges go in, and it goes down the length of the axon, and that's called an action potential, also known as a nerve impulse. If you have myelin, by the way, all of this happens faster because instead of the propagation happening down the entire length of the axon, uh, the, the sodium channels can only open between where the myelin sheaths are at the nodes of Ranvier. And so it makes for more efficient conduction of the signal. Okay, repolarization. That refers to what happens after the action potential has already passed a spot in the axon. If you leave the axon depolarized, nothing will ever happen in that axon ever again, and that neuron is now basically functionally dead. If you want to be able to reuse that neuron, you have to resensitize it. You have to make it irritable again. And so you have to repolarize it and bring it back to its resting potential. And so that's what the word repolarization refers to, how the neuron resets the axon membrane after an action potential passes through so that it's ready for the next nerve impulse. Here's how that happens. You remember that because of depolarization, we had a bunch of sodium ions that rushed into the cell, and now the inside was positively charged. You could wait for the sodium ions to just eventually get pumped back out again, but that would take too long. It's inefficient and it makes your reflexes much slower. So if you want lightning quick reflexes, you have to reset the outside of the membrane back to a positive charge. And the fastest way to do that is to just release the potassium ions. Because remember, the potassium ions were at a high concentration inside the axon. So when this orange potassium channel opens, the potassium ions rush out, and now the outside of the axon is positive again, and the inside gets to be negative again. Here's another depiction of that at the bottom of repolarization. So you have these red dots which represent potassium ions, and when the potassium channels open, the red potassium ions rush out, making the outside positive again, until eventually the channels close and the potassium gets pumped back inside the axon where it belongs. I would recommend that you pause the video at this point and just compare depolarization on the left, which is also shown in the top panel in green here, and compare that against repolarization on the right, which is also shown in the bottom red panel down here. And just make sure that you know in your own mind when can you tell when an axon, or how can you tell whether an axon is depolarized or when it's repolarized. Okay, last of all, at the end of repolarization, you're going to have a lot of potassium ions outside and sodium ions inside, which is backwards, right? Like on the one hand, potassium ions rush out, and yes, we have a positive axon, a positive exterior to the axon. But those potassiums really belong inside the axon, and those sodiums really need to go back outside the axon to get us back to resting membrane potential. And so that's where the sodium-potassium pump comes in. It's a protein in your axon membrane that's going to utilize ATP to pump 3 sodium out and to pump 2 potassium ions into the membrane. And I want you to notice that ratio. 3 sodium went out, 2 potassium came in. So there will always be more sodium outside the cell than potassium inside the cell because of that ratio of 3 to 2. So you can see here the sodium ions inside the cell will occupy this protein pump. ATP comes in and changes the shape of the pump, which allows the pump to dump the sodium outside, while at the same time allowing potassium to come into the pump. And then we're going to change the shape again, which then dumps 2 potassium inside, and that's how we get the sodium to go back out and the potassium to come back in. So here's all of that in words. Once the action potential or the nerve impulse starts, it's propagated over the entire axon. And this is an all or nothing principle. Either the action potential goes all the way down or it doesn't. And if it doesn't, then you just have no nerve impulse. And so there's no such thing as starting an action potential and then having it like, wither down to nothing. That doesn't happen. Either it starts and gets all the way down or it never starts at all. So once the action potential starts, it's going to pass all the way down to the end of the axon, which is shown in panel D. And then next, potassium ions will rush out of the neuron, which repolarizes the membrane. And so that's shown in panel E. What actually happens is the membrane will temporarily become hyperpolarized. Hyper meaning overpolarized. So the outside, because so much potassium goes out, the outside becomes actually too positive, and the inside becomes a little too negative. But don't worry, the sodium-potassium pump on the membrane is going to restore the original configuration by putting the sodium back out and pulling the potassium back in. Here's a graphical representation of what happens during a nerve impulse. You are required to know this graph. It will appear on your unit exam and probably on your next daily quiz or quiz as well. So you have membrane potential on the y-axis, meaning the electric potential, and you have time on the x-axis. Right here, this is a neutral electric potential. Anything above it is positively charged. Anything below it is negatively charged. So resting membrane potential is where the inside of the axon is negative. But when an action potential happens, as long as you cross this threshold, as long as you cross this threshold line, you're going to get a quick depolarization as sodium ions rush out, and then a depolarization as sodium ions rush in, repolarization as potassium ions rush out, temporary hyperpolarization because too much potassium will exit, but then don't worry, from this point on, the sodium-potassium pump will restore the membrane back to its resting membrane potential. Okay, so I'm going to go through that again. We're at resting membrane potential, a stimulus comes, sodium channels open, and sodium ions rush in. Now the membrane is positive. Now it's time to reset everything. And so potassium channels open and potassium ions rush out, temporarily hyperpolarizing the membrane. And then right here is when the sodium-potassium pump gets to work and is pumping the potassium back in and the sodium back out to restore everything to its resting membrane potential. Next, what happens is synaptic transmission. So once the action potential gets to the very end of the axon terminal, it's going to now go from that neuron to another neuron. And so here you can see we're at the end of the axon. Here's the axon terminal. Here's the dendrite of the next neuron, and there's this little gap right there called the synapse. And so what's going to happen? We're going to release these chemicals there. They're going to cross that gap, and they're going to start a new action potential. So here's a little GIF of that. We've got these neurotransmitters, or these chemicals called dopamine. And when the action potential comes, that was that flash of light, they release the chemicals to the other side, triggering a new action potential. And so that's going to be represented by that brief flash. You can see the structure of the synapse, which is the place where an axon terminal of one neuron connects with the dendrite of the next neuron. And you're going to learn more about that in a future lecture. Okay. I'm not going to read any of this, but if you'd like to, you can pause the video at this point and you can read through it briefly. And then last of all, you do need to know the word neurotransmitters. So neurotransmitters are chemical messengers that transmit action potentials from one neuron to another across those gaps between the neurons, which are called synaptic clefts. Here's an example of what can cause neurotransmitters to not work the way they should. And here are some common neurotransmitters that we're going to be talking about at some point throughout this unit. Here are your key takeaways. Neurons transmit nerve impulses to communicate with other neurons, and the resting membrane potential is what enables neurons to be both irritable and conductive. Second, nerve impulses occur when the neuron membrane is depolarized. Depolarization triggers action potentials that propagate down the axon all the way to the axon terminals, and potassium channels and the sodium-potassium pump then repolarize the membrane, returning it to its resting membrane potential so that the neuron is irritable again. And last of all, a neuron transmits an action potential to other neurons by releasing neurotransmitters into synapses, and the neurotransmitters depolarize the dendrites of the next neuron. And that's the topic of the next lecture. At this point, I'd like you to go back to the beginning of this lecture, to the very first slide, and see if you can answer this essential question using all of the vocabulary words in the left column of this slide.